is Curtis Stone, and we're here today to talk about mitosis. Now, what is mitosis? Mitosis is the process behind cell division. Now, I don't know if you've talked about cell division in your classes, but here's how we're going to demonstrate it here today. Take a cell right here, and what will happen in cell division is that it will produce two new cells. Basically, this parent cell, and through mitosis, will result in two new daughter cells. So again, mitosis is the process by which one cell creates two new identical cells. And mitosis, the important thing to remember about it is that it's split into 5 parts. IPMAT, I-P-M-A-T, or if you want to remember it, I passed my anatomy test. And the 5 phases are I for interphase, P for prophase, M for metaphase, A for anaphase, and T for telophase. And the thing about these 5 phases is that they go in a cycle. That is, after the 5th phase, telophase ends, interphase starts immediately in the next cell. Alright, now we're going to talk about interphase. And to demonstrate interphase, we have an example cell right here. This right here is the cell membrane. This white little part here is the nucleus. This little ring over here is the nuclear membrane, and inside of the nucleus, as you can see, we've got the DNA, or the deoxyribonucleic acid, which is exactly what tells your body how to function. And in the nucleus, during interphase, the first step of mitosis, the DNA is arranged into these strands called chromatin. And what happens during interphase is that this chromatin, or your DNA strands, replicate. So by the end of interphase, what we have is double the strands for all of the DNA that you have. So remember, the important thing about interphase is that by the end of it, you have twice as much DNA as you started with. Alright, now we're ready to talk about the second part of mitosis, P or prophase. Now, we already know that all of the DNA in the nucleus is replicated, so what happens now is that this DNA, all of these DNA strands called chromatin, begin to condense into a form known as chromosomes. As you can see, these new forms, chromosomes, have got 2 strands, so we can go ahead and replace all of these strands here with these new chromosomes. And the next thing that happens during prophase is that the nuclear membrane begins no longer is visible. It begins to dissolve, and what happens is these chromosomes are spread out throughout the cell. And now you're probably wondering what these buggers are. These are centrioles, and they will be later involved in the splitting of these chromosomes for the next part of mitosis. Alright, now it's time to talk about the third step of mitosis, M for metaphase. And what happens during metaphase is that all of these chromosomes begin to line up in the middle of the cell, something like this. They line up in a straight pattern like this right here in metaphase. And again, these buggers here, these centrioles, what they do is they release these spindle fibers, and they attach to each one of these chromosomes on both sides, as you can see as I'm doing here. So these chromosomes are lining up as they are attaching to each one of these spindle fibers, and what will happen is in the next step of mitosis, we're going to talk about how these split for later cell division. But right now, metaphase, remember, with the key things to remember are that they line up in the center of the cell, and also that they attach to these spindle fibers. Alright, now it's time to talk about the fourth part of mitosis, A for anaphase. Now, during anaphase, what happens is all of these chromosomes that have been lined up in the center of the cell begin to split. As we can see, we got these centrioles here, all lined up with each of these chromosomes, and what happens during anaphase is that they finally begin to pull apart, as you can see right here. Now, when they were together, we call them chromosomes, as you can see down here. But when they start to be pulled apart, each individual strand here is called a chromatid. So, during anaphase, all of these chromosomes are split up, and these spindle fibers begin to pull them closer together to each side of the cell, as you can see here. So that's what happens there during anaphase, is that the chromosomes are broken up into chromatids, and they begin to be pulled apart to different sides of the cells. Now it's time for the final phase of mitosis, T, telophase. We've been through the other 4 phases, and now it's to see how this all comes together. Alright, so what happened during anaphase, the last phase, is that all of the chromatids begin to split to either side of the cell. And as you can see, all of the chromatids are now either one side of the cell or the other. Now it's time for the actual splitting of the cell. As you can see, the cytoplasm, or the cell membrane, is beginning to form a cleavage furrow because it's getting ready to form together and split apart. And this is called cytokinesis, when the cell membrane, when the cytoplasm itself is splitting into two parts. And also what happens in telophase is that on each side of the cell, a new nucleus starts to form. So all of these chromatids begin to once again form into these new nucleuses on either side. And what happens by the end of telophase, as you can see here, we've got almost two new cells. What happens by the end of telophase is that this cleavage furrow comes together and eventually they will split apart and you've got two new cells. Alright, now we're completely done with mitosis. We finished all 5 steps and it's time to see the product. Here we've got 2 cells, completely good as new, and if we look over here back to the diagram, we can see that this was the final result that we had predicted from the beginning. We started off with 1 parent cell and we ended up with 2 completely identical daughter cells. These 2 cells are completely identical to each other, and while they may seem small right now, we have to remember that telophase just ended, and so now we're back at interphase where the cell is going to go through growth and again going to go through replication, and each one of these cells is going to go through mitosis again. So that's the process of mitosis, folks. I'm Curtis Stone, and we'll see you next week when we talk about meiosis.